So thanks everyone for showing up. Let's start with that. I'll try to get the presentation done in roughly 20 minutes. Whatever I'm going to share with you now is the result of my PhD. So I'm finishing up my PhD at the University of Amsterdam, which was in collaboration with Liverpool John Moores University, and my research was funded by WWF the Netherlands and the University of Amsterdam. So I really just use that as a basis for this talk, but I have more knowledge than what I'm just sharing here. So I'm just trying to keep it brief. So this is me, obviously, on the picture, and the little silver thing I'm holding is a MinION. This is a portable DNA sequencer, and I'll talk more about that. So just to get started, this talk's going to be about genomics. And for me, in this talk, the definition of that is basically how we can study DNA to get the information of DNA on our computer screen so that we can analyze it and compare it to other databases, things like that. As a starter, I think it's useful to say that I'm assuming a basic knowledge of biology and molecular biology for this talk. However, I shared a glossary, which is in the notes that Ellie just shared. So if you're not really familiar with molecular biology, or I say some terms that you might not be aware of or not familiar with, check the notes. And also, there's also the slides in a Google slide document. So if you want to look along with the slides, you can do that. And in the slides, I also added a few slides of terms that I might use that might confuse people. So be sure to check those out if during the talk you're a bit confused. But I'm not going to show the slides here to keep it brief. Cool. So I'm studying genomics, and to give you a little bit of background, what my PhD was about is we were interested to look how portable genomics can be used for the health monitoring of great apes. So there are certain threats that threaten great apes, and all species of great apes are endangered. And for example, Ebola has been a really big problem in the past for local populations of apes. So we were thinking, like, if we can have portable genomics, maybe that can help us to identify how diseases spread, or to just monitor the health of endangered species. However, as you can see on this picture, the health monitoring of, for example, Ebola has to be done with a lot of personal protective equipment. It's really hard. You have to be very, very cautious. And then if you have the samples and you have to analyze them, you have to do it in high biosafety level labs. So that just requires a lot of logistical challenges. What I did in my PhD is we kind of took nematodes as model organisms. Nematodes are often, and there's microscopic worms, and they can often be present as parasites in wildlife. So they can be collected without having to touch the animal, because we can collect them from poo samples, and the processing of the data and the samples can be done in a normal lab. So it just makes it easier to work with. So the whole talk will kind of be based on this, but what I did was basically species identification. So if that's something you're interested in, then a lot of these things will be directly transferable to what you're doing. So the basic pipeline of data collection is you go into the field. If you are someone like me, you look for poop, and then you collect that and you store it. You prepare it for logistics. So you either, if your lab is in, for example, your capital, it has to be transported there. If it's in a different country, it has to be transported there. And then once you get your samples in the lab where you want to do your molecular work, you have a full molecular lab that has a lot of tools, big bulky tools, very expensive tools, and you do your analysis there. And traditionally this takes months if you're lucky, and years if you don't have the luxury of a PhD to just focus on processing these samples, and you have to write grants and scientific papers and all that on the side. So in my PhD, we were kind of thinking like, okay, but what if we can eliminate that logistical step of transporting the samples to a different lab or location, and we can do the genomics in the field immediately. So then it would look something like this. You collect your samples, you store your samples, you prepare your samples, and then you, for example, do the sequencing, and this can then be done in days instead of months. So for my, and then you can repeat that cycle. You know, you get a few samples, you process them, you go out, get some more samples, process them. So it would be just a continuous process. So in my PhD, I focus mostly on DNA sequencing, as I think it's a very universal method that can answer a wide array of questions. And also, I'm really interested in using non-invasive samples. For example, if you're working with wildlife, it's a great way of not making them stressed by just collecting fecal samples. And in this presentation, I'll talk that we can do the sample collection, DNA extraction, we can perform PCR or qPCR in the field. And for some applications, that's sufficient. Like some people are already just like, okay, I just have a PCR protocol, which tells me if some animals are male or female. So then you can stop there. But we're going to continue with that. I also include the sequencing and the bioinformatics in this talk. So all of these things, we're going to talk through how you could do that in a field setting. So the reason why we can now do these steps in the field is because in the roughly last 5 years, a lot of the technologies have been miniaturized. So now you have portable molecular labs, you have qPCRs that are portable, you have sequencing machines that are portable, and there's even a company that's making a fully portable lab workspace. And this has I'm working on a website which is called portablegenomics.org, and like all the details of the technologies are stated there. But right now I'm just giving you a case study of what I used. And there is a variety of researchers that have already used these portable technologies in a variety of settings. So that some people have done off-grid expeditions. They've been used for outbreak surveillance. I worked on parasites. Some people worked on plant pathogens. It's used for sex typing, species identification, and they're also very suitable for teaching and capacity building. So also this research is highlighted on portablegenomics.org. So I'm not going to go into detail of that now, and I'm just going to use my PhD research as a case study of if you want to do DNA extraction and sequencing, then what do you actually need? So this is the basic portable setup that I used, and this is how we used it in Indonesia. So last year I went to Indonesia and I trained some veterinarians there in these technologies. So in the back, so you see the laptop, and in the back of the laptop you see an orange and white thing. That's a bento lab. This is one of the portable molecular labs that is available. There's another company called Mini PCR who also has molecular biology labs present. And then on the right of the laptop you see something called a Minute. That's like a mini computer that's associated with the MinION, which I showed on the first slide, the silver thing next to it. Then we have a laptop for data analysis, and on the right top you see a magnetic tube rack which you need for the sequencing, and this is a handheld tool on the right which I used as a vortex to mixture samples. So this was basically the basis of my portable genomics lab, and I'm just going to show you how we use that. So we managed to perform DNA extraction with the bento lab. One of the things you need to do in a DNA extraction is lyse the cells to get access to the DNA. And usually you use a heat block for that, but the bento lab has a thermocycler. So what I just did is instead of leaving our solution in large tubes, we transferred them to 0.2 thermocycler, 0.2 milliliter tubes, and put them on the thermocycler at like an indefinite time, just so as long as you would normally put your samples in a heat block. And then there's a centrifuge which has tubes, and it can just hold 1.5 milliliter tubes. But if you just put some tubes without a cap in there, you can put your thermocycler tubes in there and spin them down. So the bento lab centrifuge, you can use it for just spinning down of samples, but you can also use it, you can also change the centrifugal forces. So if you need to run something at 8,000 Gs, you can. And then, like I said, the handheld tool was a way to vortex and shake the samples. Then we prepared the PCR. So obviously we didn't have a PCR like workspace in Indonesia. It was a veterinary clinic, but we just put some aluminum foil on the table. We cleaned that with bleach and ethanol to try and have like a kind of a clean workspace. And then we prepared the PCR and then put it in the bento lab and did our normal PCR. And then to see if our PCR worked, we performed gel electrophoresis. So normally I melt my gel in a microwave, but we didn't have a microwave there. So we just bought a camping stove and we used a traditional coffee pot to melt our gel and then pour it in the chamber. And then the bento lab, as you can see, has like this chamber that you can make your gel in. But it also has the blue thing underneath is a transilluminator, so it has a light with which you can show your bands. And I'm not sure if the bands are clear enough for you on your screen, but there are some light bands on the lower right picture to show that, yeah, everything worked, the DNA extraction, the PCR, and the gel electrophoresis just gives good results. And then after that, we also use the bento lab for the min-ion prep, and because you have to prepare your DNA for sequencing. It's called library preparation. So all these steps were also able to be done on the bento lab. And then we did the sequencing. We put the DNA on the min-ion, the sequencer. And then this is how happy you look after you've done hours of work, and you're really happy that something is actually generating biological data. So it was really cool to see that that worked. And then you have your data, and then you have to do bioinformatics. So this is a very complex figure. I'm not going to explain it, but basically the authors of the ON-track pipeline, they say that their pipeline is intended as kind of a Pokédex for the min-ion data. If you're familiar with Pokemon, you know what I'm talking about. But basically what their intention with the pipeline was, you have your data, your sequencing data, you put it in the pipeline, and then the pipeline runs a certain amount of steps, does some quality control checks, and then gives you as an output a consensus sequence, it's called, and then you can compare that to a database. So that's what the bioinformatics was that I use, and it's quite straightforward. And then this is the kind of results you can get. So with I did my first paper is just a proof of principle with 4 species, and we got 99% to 100% accuracy for 3 of the 4 samples, and a slightly lower accuracy for the 4th sample, which was probably due to the flow cell being quite old. But yeah, that was really encouraging that we can get such high accuracy compared to standard sequencing. That was all just kind of showing like what you need to do, how you can do it, and that the results can be quite accurate. And then obviously I'm giving this tutorial because I think that there's a lot of advantages to this system, and I think other people might benefit from this. So the first one obviously is that it's portable. You can do molecular biology at atypical sites. Everything I needed for a month of training in Indonesia fits in one suitcase, one normal standard suitcase. It was a bit heavier, but not much bigger than a standard suitcase. And I think that's amazing. Also, another advantage is that it has lower startup costs than traditional molecular equipment. On the website, I'll talk a bit more about it at the end of the talk, but I also tried to specify an estimate of prices, and if you would compare that with like traditional equipment, you can see that it's much more affordable. So I also think it might be good that maybe this brings molecular biology to non-standard institutions that might have less financial resources. Another advantage I think is that these technologies are quite straightforward, so they will require less maintenance or less servicing of the equipment. I know of some stories of really fancy equipment ending up in remote labs, which is great intentions, but then they need servicing, they need a lot of training to understand how they work, and then they are just not used. So I think that's a shame, and these kind of technologies are, from my understanding, quite straightforward and less expensive. So if you do not happen to not use it for 6 months, it's not that big of a loss. And from my personal experience, you can perform the same kind of experiments on these portable tools as on traditional molecular equipment. Anything I've tried so far worked on that as well. So that's also really good. Another main advantage, which I already mentioned in the introduction, is that the time investment can be quite short. So in my case, with nematode samples, the time investment from sample to species identification can be roughly 3.5 days. There are some caveats. That's obviously because now I'm very experienced in both molecular biology and bioinformatics. And this is, you can watch the slide later if you want to get all the, want to know how this is built up. But basically, it really depends on what samples you're working with, what kind of kits you're using, what's your research question, what kind of bioinformatics you want to do. So this is really just based on my research, but it's a good kind of estimate for how much time, if you would be working full-time 8-hour work days, in roughly 3.5 days, you could get your answers. So the main time I want to spend on this is to talk about the disadvantages and potential bottlenecks, because I feel like that's probably what most people are curious about, and also how they can learn from, for example, people like me or other people that have done this already, so that they can prevent some mistakes. I think the main point I want to make here is decide whether you need genomics to answer your question. It's really cool tools and it can be very useful, but there are some things that you might just not need genomics for. And just be critical when you plan your field experiments, when you plan your research, to really decide, do I really need this? Because maybe you can get similar quality information with different tools. And one of the reasons why I say this is that molecular, genomics still requires quite good molecular biology skills and basic necessities. So you need people that know how to pipette, how to do gel electrophoresis, PCR, all these things. You need personal protective equipment, you need pipettes, pipette tape. So it's not just something that you can do without a lot of putting a lot of thought into. And another thing that comes on top of that is when you're in the field, you need to think about proper waste disposal. Normally you're in a molecular biology lab and you have like your yellow little bins and stuff will get burned and things like that. But if you're in the field and a remote location, this is really something you have to think of yourself. And like the point I was making, for example, if you can get some of your information with microscopy, it will still be cheaper to do microscopy than it will be to do genomics. You will get less quality, high quality data, for example. You won't have the genomic data, but sometimes it might be sufficient. So I don't want to advocate that everyone should replace their traditional methods with genomics. Just think about how it can complement the research you're doing. And even though I just said that the time investment is an advantage, I also think there's a disadvantage, or it can be a potential bottleneck. Like, it will take time to get used to the miniaturized equipment. It will take time to figure out how to adapt your protocols to the miniaturized tools. It will take time to get highly skilled at doing genomic, which means getting fast at it. And it takes time to get good enough at bioinformatics. So just be aware of these things. Some people see the MinION and think, oh, great, I'll just buy it, and within 10 minutes I have all my answers, and that's a bit too simplistic, unfortunately. Another bottleneck for field research is that it requires electricity to run genomics tools. So you can overcome this with battery packs or solar panels or generators. But sometimes if you have to bring in stuff by plane or import things, then, for example, lithium-ion batteries can, there are restrictions on how you can bring that in a plane, how big they can be. And also solar panels can be great, but if you're in a rainforest with lots of cloud cover most of the time, then solar panels don't always offer reliable electricity. And I mean, you can have a generator, but if your generator is used to run the whole camp and you only have to use some electricity for your sequencing, then you're obviously not going to spend a lot of money on gas just to run a small experiment. So these are kind of things you should think of. So battery packs work, car batteries can also work. We got that to work in Indonesia, but then still you need to think of, even if you use batteries, how are you going to recharge them? And then that brings us to refrigeration. Molecular biology often uses reagents that need to be stored in fridge or in a freezer. And if you don't have that, then there are a few things you can think of. So, for example, you can choose DNA extraction kits that can be stored at room temperature. I used brought two different kits to Indonesia, and one required some reagents to be stored in the freezer, but the other one, a Keating kit, everything could stay at room temperature. Another option is to buy lyophilized reagents. So it's freeze-dried reagents. They can have quite long shelf lives, months, or I found an internet link that said up to 2 years. So I believe them. And I know that there are a lot of companies that sell these kind of stuff, lyophilized. So Oxford Nanopore Technologies, which is the producer of the MinION and the sequencer, they have a field kit, which is lyophilized. Primers, usually for PCR, some PCR reagents, you can get them lyophilized, which means that, yeah, you don't need to put them in a fridge or a freezer. So that can be a solution. Some field sites, like the one in Indonesia, they did have a fridge or freezer available, but electricity is intermittent, so it's not very reliable. And then it can just simply help a lot to leave them closed as much as possible and fill the freezer and fridge with ice packs to bridge gaps in electricity. So only open the fridge if you've got electricity, and if you don't, then just be careful so that the cold stays inside of your fridge. And otherwise, there's something called a Credo Cube or high quality cooler boxes. So if you have a short-term stay and you're just going to go to an expedition or something like that, then cooler boxes with ice packs can be plenty sufficient. Like from my own experience, I can say that, for example, MinION flow cells, the thing on which you load your DNA when you're doing sequencing, they're quite resistant to temperature fluctuations. Yeah. Don't worry about that too much. So another bottleneck can be, like already touched upon, the bioinformatics. It takes time, skill, and good computer resources. If you have a bioinformatician, cling on to them. I would love to have had a proper bioinformatician on my project. And otherwise, it's fine. You can do it during yourself. Like I taught myself, it just takes months. So be aware that it takes practice. A lot of the tools for MinION sequencing is command line based, so you kind of need to know how to program to do your data analysis, and that can seem very daunting, but there are a lot of online resources that you can use to get better at that. And a laptop might be enough computer power for a pipeline like I just explained, the ON track. But if you are looking to do really ambitious things like whole genome assembly, which then that takes a lot of data and a lot of computer power. So just be mindful of these kind of things. And then if you have your data, you have to be aware that your species identification, for example, depends on the quality of databases. So if your species isn't in a database, you can have perfect sequence data, but you won't get a match to the species you were expecting, simply because nobody has put the data in the database yet. So, I mean, for me, that's one of the reasons why everyone should have a portable genomics lab, and especially in remote regions, because I think if you're someone that has a lot of species knowledge and you can morphologically identify a species, and then you can do the genomics, then, we have new information in a database. So I think if you're that kind of person, know that you've got a valuable skill, and this is how we can build databases. So I think that would be really cool. I'm almost done. And then, so one of the things I kind of have been trying to explore as well, and I'm very interested in, is frugal science. It's kind of like exploring how we can reduce the costs. So for example, the magnetic rack I was talking about, if you buy that commercially, it's roughly £200, which I think is like $250 US. But by getting it 3D printed, you can reduce the cost tenfold, and by using a multi-tool, which you can just buy in any hardware store, and using that as a vortex, you only have to spend like £50 instead of £250. So these are kind of things that you can take into consideration. And then this is what I was talking about at the beginning, portablegenomics.org. I've really tried to get the link live before this talk. It's not really quite worked. I did send the temporary link to Ellie, so it's in the notes, but this should be live hopefully tomorrow. And there I've made an overview of all this portable tech with price estimates. There's a glossary for if you're new to genomics, some educational resources and some of the highlighted research of other people that have done this in the field. And yeah, I would appreciate your input for that. So what kind of information would be valuable for you to see on there? And if you're interested in contributing, then just let me know, contact me through the website or on Twitter. And then that's it. And I'd love to answer any questions that you guys got.
Awesome. Thanks, Ineke. That was super interesting. Just before we get into questions, what sort of contributions would you be interested in for?
Yeah, that's a good question. So one of the things I'm thinking of, first of all, is questions. So like if people like are interested to know, like, for example, this week, the reason why I included the refrigeration in here was because I got an email from a scientist that was interested in that. So it had kind of slipped my mind. So this is obviously very useful. But also I'm thinking if there are people that have worked, like the field site I was in at Indonesia was still quite luxurious. There was a building, there was electricity. So if there are people that have worked on more remote places and yeah, I would appreciate input of maybe some logistical challenges they overcame that we can share with other people, but also their kind of day-to-day hacks, just tips and tricks that people that do a lot of field work have generated, and especially, of course, if they have experience with genomics, but even otherwise, like battery packs, solar panels, things that can help portable genomics. If you have any tips or tricks, then that would be, I think, very valuable to include on the website.
We might put a call out, like that specific call on WILDLABS in our eDNA group and in your thread as well, just to make sure that call is there so we can work with the community as well. Really lovely feedback in the chat. Carly, I think you're up first. Do you want to jump in?
Hi. Yeah, this is awesome. We work in Madagascar in the middle of a rainforest where, yes, a lot of these problems apply in terms of refrigeration and electricity and sample storage, and exporting samples from Madagascar to the US is just a nightmare process. So it would be great if we could do this all in Madagascar. I just wanted to, do you have like a range of just cost for like this whole process? Like if we're starting from, you know, generally, like we have a genetics lab in New York, so, and like there are a bunch of people in our lab that are like good in this, you know, like processing and PCR and all of that kind of stuff. But if we want to like set it up, you know, what's like the overhead gonna be to get this whole process?
Yeah. Yeah, so this is initially I had dreamt of including all the price estimates in this presentation, but as you noticed, even when I tried to keep it to 20 minutes, I went way over. So a very good question, and this is why I've included all these prices on the website. But I've also written I'm co-writing a review now on new technologies for primate conservation, which will be out hopefully later this year. I also mentioned some research. So there are a few articles that have mentioned price estimates of their setups. One of them is in Madagascar, which they worked on lemurs, and they have like quite a basic setup. And I think that their whole lab was like $2,000 US dollars, but that was excluding the sequencing. So that's if you want to do DNA extraction, PCR. And then it kind of ranges between that and like the highest price I've seen was 10,000 US dollars. But that is that they were trying to do more high throughput sequencing and processing. So one of the things that you are, of course, with the miniaturized labs have is you can just process less samples in the same amount of time. So like a bento lab is roughly, let me just quickly peek because I have it open here, because I'm trying to translate it to US dollars because I think that is more like universally understandable. is the one with full functionality is roughly $2,000. And if you get mini PCR portable, full portable molecular biology lab, that's roughly $1,500. So that's the DNA extraction PCR kind of part. Sequencing, if you've got a normal MinION, which that is $1,000 for a starter pack. They also now have, so in my presentation, I showed that there is, I had a mini computer attached to the MinION to do the processing. So now they also have a new tool that you can combine everything in there. So you have a sequencer, a mini computer, and a screen to see the analysis, and that is roughly $5,000. So this is, with the MinION, that's kind of then your starting costs. And then you have to include flow cells or something called a flongel. So that's your kind of cartridge for your DNA. And they go from, a flow cell can be $900 if you just buy one, but they get cheaper if you buy them in bulk. You can do multiple samples on that. It's like, for example, you can easily put 12. If you're doing what I did with just amplicons, you can put way more on that. You can do, they have 96, yeah, up to 96 samples. But they also now have a flongel, which is a smaller DNA cartridge and is more for like PCR, sequencing of PCR products, and that's $90 per sample. So long story short is, if you get, for example, the Mental Lab's £2,000, you get your MinION is £1,000, and then that's assuming you already have pipettes, and then you need a good quality laptop, maybe another £1,000. So I would say for $5,000 dollars, I mean, $5,000 you should get quite far. Yeah, I hope that. And but I will, like, one of my aims is to get more of these kind of like estimates also on the website. So just, yeah, keep an eye on that in the next few weeks and months, because I do have some of these examples calculated also. For example, the price for the reagents per sample for sequencing, but it's a bit a lot to consider.
Yeah, no, thank you so much. That's very helpful, and we'll definitely keep an eye on that. Yeah, I study lemurs. Were you in, was that lab the Mahaliana lab?
I know that, I only know the name of the first author was Guevara, if I pronounce it correctly. So I can send you I think the study is one of the studies I linked on the website, and there's a temporary link to the website in the data file.
Okay, yeah, awesome.
All right, Rob, you're up next. Do you want to jump in?
Yeah, thank you so much, Ineke. That's amazing stuff. So you might have mentioned this right at the start of your talk, but I thought you said you can potentially sequence all the way down to individual level. Is that possible?
Can you specify what you mean with all the way down to individual level?
So if you found, so I study dingoes a lot, or koalas, for example, and if I wanted to know individual, like if I wanted to do DNA mark recapture, would I be able to get sequences that would be able to tell me down to individual koalas or dingoes?
I wouldn't know why not. So what kind of samples are you working with? Are you working with fecal samples, or are you working with like sometimes blood samples or things like that?
I think ideally we would work with fecal samples.
Mm-hmm. So I think for me, this is definitely one of the things I'm also really interested in. I haven't tried it out myself yet. I know some people are working on that as well, but I do know there's lots of people that have successfully isolated DNA from individual animals from a fecal sample. I know that there's a lot of people in grade 8 biology that do that. They try. And then I also know that if the quality of your DNA is good enough to get, either an amplification of the individuals, or you get a good enough DNA quality, then you can just sequence it. So my basic question, the short answer to your question is I'm not 100% sure, but based on what people have done so far, I wouldn't know why it wouldn't work, if that makes sense.
Yeah, amazing.
Yeah.
Amazing stuff.
And also, I have to admit, the technology I worked with in Minayan, these were like already an older generation flow cell. So I also know that nanopore technologies is quite rapidly increasing the quality of their reagents and chemistry. So that has also already, like when I started 4 years ago, my supervisor and me like, oh no, why did we start working with this? Because this looks horrible, this data. But like now we get quite accurate species identification. So I really also just believe that this is just a matter of time that this will really reach good quality and can be definitely used for things like this.
Okay, thank you very much.
Paula, you're up next. Do you need me to read out your question, or do you have a mic? Okay, good. Do you want to join in?
Sure. Sorry, I didn't see your question before. Hi. Well, I'm from Colombia, and currently we are starting to work on field-based genomics, mainly for species identification, but so now we have a project with the Andean bear with fecal samples, and I was wondering if you have any recommendation on how we should preserve these samples to avoid DNA degradation when there are very high temperatures, or maybe not, but a long wait till we get to a field site, for example, if the DNA degrades quickly. And thank you for your talk. It was very cool.
Cool. Thank you for the feedback. So yeah, if you don't mind, I'm just going to ask you a question. Like, what would be your normal way of processing? So do you have a field station from which you sometimes do expeditions to collect, for example, fecal samples? Or do you sometimes go into the forest, collect samples, and then it can be days or weeks before you actually come back to your kind of field station?
Yes, it could. There might be the two options.
Okay.
Most of the time, we do have to wait for a week till we get to the field station, because we spent a lot of time looking for samples, and then we got to the field station, and then because that's the place we set up the lab and process the samples. Yeah.
Okay, so that's it. So to be honest, that's a really good question, and it's one of the things I had hoped I could reach. One of the stages I hoped I could have reached in my PhD, to work directly with fecal samples. Excuse me, and but unfortunately, I didn't quite. Like, we always isolated nematodes from the fecal samples. So my real honest answer is I'm not 100% sure what the best way is to conserve your fecal samples. The other answer is I can find out for you, because I kind of know where to search for. So I hope this is okay if I just follow this up and take some time after the presentation to look through some sources I know that have worked with fecal samples, and then I could share some research articles with you that hopefully will answer this kind of question.
Oh, sure. Thank you so much. That's great.
We've made a note of that. And we'll share whatever you share it in the thread of this discussion. Also, when you're joining and asking a question, do you want to? If you don't have to, but do you want to turn your cameras on as well? Just because it's quite nice to see face to face who we're talking to. That's suggestion, not a compulsory thing. Anyway, I'm not sure if I'm going to say this name right, so I'm sorry if so, but Ineke, you're next, I think.
Okay. Thank you very much, Ineke. This is a very good presentation, lovely, with quality in genomics research field. Now, my question, actually, this is not my field, but I would like to ask some certain questions concerning the presentation. As regards to sample collections, I am thinking of from the tropical area where I came from, whereby the temperature is a little bit higher than where this presentation or where you are. Please, may I know the recommended temperature for sample preparation and preservation in case it's going to actually, probably the researcher is not ready to carry out the research now at that particular time, or for how long can the sample be preserved that the result will still be valid. That is one. Then secondly, another question that I would like to know, based on the tools you actually talked about, the number of samples that you can actually run at a go, and at the same time, how do you compare your results with standard? That is, what standard do you use in calibrating the non-sample? And at the same time, may I know if this lastly, which is the third question, is that I would like to know, can this tool you actually analyzed or you talked about, can they be, for instance, if I'm actually, as the case is now, like this currently, the pandemic issues the COVID-19 issue, is it possible for us to know that, oh, these particular strains of the virus in this location actually comes before this in the other locations? Can we actually use this tool to differentiate which one takes place first before the other one? That is, to just trace the origin of the disease or ailment. Thank you.
Okay, great, thanks. I took quick notes of the questions, but if I miss something, please don't hesitate to remind me. So your first question, if I understood it correctly, was, so you work traditionally in very tropical environments, and basically is the sample preparation suffering from that? And also I think the second part of the question was, how do I store the samples up until the moment that I can process them? Is that correct?
Yes.
Okay. So yeah, so we worked in Indonesia. I trained the veterinarians there, and that is also very tropical. But they had the advantage of having air-conditioned systems. I know of some researchers that did some work in the Philippines that, so first of all, I think the DNA extraction, the PCR and the sequencing itself shouldn't really pose a problem. For example, the BentoLab has a fan, the MinION has a fan. I know there are some people that do some research in swine fever, I think was in the Philippines. And because their labs were really warm, they 3D printed little standards for the MinION to just allow more ventilation around it. So I feel like, you know, sometimes even if you work in a tropical setting, making sure that you have enough ventilation space around your tools. So maybe just elevating them might be a good way of at least taking care of the technology. Having said that, obviously there are the problems with humidity. If you're working in the tropics, how I tried to prevent that was just having a box with silica beads, an airtight box. So if I was not using my tools, I would try to store them in the airtight box with silica beads. And I know there are some silica beads that you can just reset in the oven. It will take a bit of preparation, but that's one way of trying to take care of your technology in tropical situations. And then I think the answer for how to kind of store your samples until you process them is a bit related to the previous question. It depends on your sample. If you have some sort of cool box, a cooler box, or things like that, that will be beneficial. I think ideally, one of the people I talked to, he was like, oh, if we can just do the DNA extraction as soon as possible, then your DNA, if you're working with DNA, is quite stable. So then you can just store the DNA. But again, if you really are interested in sample storage solutions, then maybe we can talk afterwards or on email on like what kind of samples you're working with, and I can browse in the literature to maybe point you to some research, because that is quite a specific question, I think. With regards to number of samples, the slide I showed with how long it takes for 3.5 days from sample to identification, that was based on 15 samples. So that was 15 individual nematodes, 15 separate like DNA extractions. And the main reason is because, for example, the BentoLab it has a small centrifuge with you can just put 6 samples at the same time. So it really kind of depends. If you use the mini PCR system, you can have a mini centrifuge that can hold larger tubes, so you can kind of speed it up a little bit. But I would say for my stand, like what I just now did last week was 15 samples, and I felt that was ideal, because if I would have done more at the same time, the centrifuge really would have been a bottleneck. But if you have more samples to process, just buy 2 and put them next to each other. Or with the mini PCR, the advantage is you can, for example, just buy 2 mini centrifuges and then speed up that a bit, because you can just kind of, there you can kind of just build your own setup. So that's one of the ways you can do it. But like the main take-home message is, yes, portable genomics is possible, but it's not going to be high throughput. If you really want high throughput, you still kind of need a traditional molecular biology lab. And then the last question is, yes, it is possible to know the strains in the pandemic. There's a lot of people that use these tools now in the SARS-CoV-2 pandemic. I had intended to also mention that in the presentation, but well, there was just too much to talk about. So I'm happy with the question. So yeah, there's a lot of people that work with the MinION. In the pandemic, if you go to the website of Nanopore Tech of ONT, they'll share a lot of resources there. There is even a standardized bioinformatics pipeline, NextStrain, and there's also actually a project underway, which I'm a part of, which is called Accessible Genomics, and it's a part of Joggle, which is an open source or an open science platform where people are trying to see, like, how can we make genomics for healthcare, how can we streamline it. So the point of the project is to kind of set up for SARS-CoV to set it up in the Philippines, but then really meticulously track everything that needs to be done to get it up and running in a developing country, so that, or a remote region, so that other people can copy exactly what has been done there. So that might, if people want to know more about that, we can, I can probably find a link somewhere and we can share that. So yes, you can definitely sequence viruses and track the pandemic with that. It's happening as we speak. Yeah.
Yeah, Ross has just dropped in a link as well that might be of interest, and we'll if you can find that link, we'll add it into the notes as well. Mauricio, I think you're up next. Do you want to join in? Okay, he might not still be here. Okay. The question was, in your experience, what kind of nanopore protocol do you recommend to use for sequencing? Okay, I'm gonna check for you. Sorry, everyone. Yeah, Pascal. It was Mauricio trying to join. So the question is, in your experience, what kind of nanopore protocol do you recommend using for sequencing metabarcodes in vertebrates? I mean, with users-defined primers for target regions.
Okay. So I feel like this is, I've always used the ligation sequencing kit, and I feel like I've always understood that that's the most versatile kit for from ONT. So I would recommend that. But to be fair, I think if you really want to know and this is going to sound really boring, but the customer service of Nanopore Tech is really good. They're really fast. You can chat with them, and this is really the kind of question that they would be able to answer exactly in detail for you. So I also know that there are some papers on meta barcoding. I do know that people are starting to do it, and with these latest chemistry flow cells, it looks more likely that it will be successful. So yeah, I only have experience with ligation sequencing kit. I think that's the honest answer. I know that there are some rapid kits as well, but that's I think all I dare to say about it. Sorry.
No, that's all we could ask, an honest answer. All right. If anyone else has experience they want to share, because I know there's some other experts in here, do you want to jump in and answer that question in the chat? Otherwise, we can pick it up on WildLabs. Jonathan, do you want to jump jump in. Jonathan Minchin. Oh, it's all good, Mauricio. Sorry. Jonathan.
Hi there. Can you hear me okay?
We can.
Yes.
So thank you very much, Ineke, for the talk and for everyone for hosting that. I work with the fabrication laboratories, particularly a green fab lab. We've worked with DIY Bio before, and it's a way to try and make everything. And I wondered if you had any experience with the WILDLABS Pocket PCR. We've tried the mini PCR, which is great fun, but I think the WILDLABS looks a little bit cheaper, maybe not so high data quality. But I was interested in your experience with using any of the WILDLABS kits.
Yeah, it's a good question. So I noticed while we were starting this presentation that because the picture on the announcement is a WILDLAB original, and I think it was credited to me, but it's not my picture, I have to admit. So I have not actually worked with it, but what I have, so for anyone that hasn't heard of this, WILDLAB, I think it's called micro PCR, is really, really tiny. And I can be like this, but on your screen it will look however, like whatever screen you're looking at. So that's very useless. But basically, it's all completely open source. So people, I think it just has bill of materials. If you buy the materials, you need to program something, but you can just build your own. There are other open source PCRs. I know Chai has an open. It's called OpenPCR. Which is cool if you're really good at making stuff. I'm better at using this stuff, but so I don't have experience with this one. I'd love to be able to make it one day and actually use it. And you say I don't know how good the quality is. If it's a normal PCR, I know that with qPCR it's a bit more sensitive, but with a normal PCR, I wouldn't really know why it would really be lower quality. It mostly depends on how quickly it can switch from temperature, but like the technology is it's so good now that I expect that that's still quite reliable. So yeah, I think I've only heard of that, and I would say that I have confidence in these kind of things because I just feel like it's quite straightforward and the technology is just getting better and better. So I don't know if that was an answer to your question.
Yeah, that's great. Thanks.
Okay.
Jonathan, it looks like there's Jingwen in the chat has used it, so you might be able to talk to each other for experience there. Okay, Naomi, I think you're up next. Do you want to join in? 2 questions left.
Yes, hi. I'm from Mexico, and I'm interested in the biodiversity on islands, and it's not easy to work on there. And it is possible to use this process for the environmental samples like soil or water. We are interested to discovering and monitoring the bacteria species. But sometimes it's not easy to manage or to take the samples, and we lost several species when we change or we take the sample soils because several bacteria is degraded or its diet. I don't know, is it correct? And I am interested to use this kind of the technologies, but I don't know if it's recommended.
Right.
What do you think?
So I definitely know that there are some people. So first of all, there's a lot of people doing 16S sequencing with the MinION. So that's definitely something that's happening. There's also more and more people that are looking into using the MinION for eDNA. So that's what you're talking about, environmental DNA. I also know that part of what that is sometimes metagenomics, and that is still a bit challenging in the MinION. People are working on it, and I feel like this is also kind of going to be related to this increase in chemistry and technology, like the improvement of that over the next few months, years. Because basically one of the downfalls of the MinION was that the error rate was quite high. It's, like I said, with the improved chemistry, that has massively reduced. But for metagenomics, where you can have a lot of samples that are quite closely related, if you have a certain amount of error, then you will just see two species as one species. So that's why for metagenomics, people are starting to look at it, but there are people with more experience that will know exactly where that is right now. There is a review, though, that I can share that is looking at these kind of questions. And so I will also share that one, and you'll get your hands on that. So because they basically, it's a review of what has been done so far and what kind of technological advances do we need to get there. So yeah, I think the easiest answer again is that I'll curate a few research articles for you to get started with, and then you can just browse through that and ask me any personal questions if you want.
Naomi, I've also dropped in a link to, we had a virtual meetup, had 3 speakers around eDNA recently, so that might also be of use. So the link's in the chat for that. Ian, I see you're already ready. Off you go.
Hi, Ineke. It's good to see you again.
Nice to see you again.
Congratulations on the work, and thanks to the other people facilitating, doing a really good job. I asked you this question a while ago when you were here, but I'm really interested in the use of this stuff to identify songbird species and subspecies. There's a lot of interest in setting up captive breeding programs for pure strains of not just like Aceh subspecies, races, or, as they call them, but also some of the offshore islands now. And the genetics guy who's sort of into this stuff, Frank in Singapore, he's a real splitter. He's not one of these guys who keeps species together. He wants to split everything into 100 pieces. So I'm really curious about whether we would be able to use this to go up to a dealer, a market, and if we suspected a minor bird was from Nias or a Murai Batu was from Simalu, whether we might persuade the owners to give us a sample, and whether this would help us to distinguish species, probably not, but more subspecies and even races. And if we could, what kind of sample would be the minimum that we could need? I mean, these guys are owners and traders. They don't want to have prime wing feathers ripped out of their wings. Would fallen feathers at the bottom of the cage, would shit samples, would tiny 1 millimeter clippings from the end of the toenails be enough? What's your thoughts on that?
Yeah, great question. So in essence, I feel like if there are 2 parts to the question, I think. So part of it is species identification, and I think that stands or falls with building a database. So if you have known species, like purebred species, and you're going to build, you have to, I think the start would be to build a local database of like, which species do you have? If you know that there are some that are pure, then, use those as reference sequences, so to say, to compare other birds to. And another part is that I know some people are interested in geographic origins. So like, can you assess from which region a bird would be? And I don't know, is that something that you guys are interested in, or is it more really the species?
The goal is to try and preserve or prevent the extinction of these various different races. But to get the birds, we need to buy them. Right. Some of them are not illegal, so we can't just come and confiscate everything. So we have to buy them. And we don't want to buy them if we don't know it's not a pure Simalu charmer, or it's not a pure Nias minor bird.
Yeah. So the main thing that I'm thinking of is what I used to work with birds in my undergrad and part of my master's, and what we did a lot with these kind of birds is, and it would be the best quality sample, is you have some veins in the wings of the birds, and if you just like prick with a little needle, you have a little glass tube, you can get like a drop of blood, and you could use that for DNA. I think that would be probably, it wouldn't require taking any feathers. I know that there are some people, I can share an article with you of some people that have done it with non-invasive samples and have tested it for, well, not for birds, but for mammals, what kind of non-invasive samples and also what kind of DNA extraction would be best. But blood is the best, and in the case of birds, also one of the easiest, because especially if they're birds in cages, they can be handled, and then you just take a drop of blood. So I think that would be the way to go. And I can't. And if the sellers are also okay with you approaching the birds, then they don't. Yeah, exactly. You don't have to buy the birds. They don't have to lose any prime feathers. And the bird, this is such a tiny prick that's restores in like seconds. So I would say that would be the best.
My last question then is, the minimum so far is 3.5 days to get a result.
So no, so this is also, this really depends. Part of this is that it's based on 8-hour workdays, but also it's based on nematodes, and nematodes are quite hard to analyze, and this was based on if you want to do an average of 15 samples. So if you, in your case, I would assume that you would go out to the market, would find 1 or 2 birds, maybe 5 max, but I don't assume you want to look at all the birds in the market at once. And then blood is quite good to get DNA from, so I think the DNA extraction will be much faster. It will probably be, instead of a day, it will be an hour or so. And then you would still have to do a PCR, 3 hours. You would have to do the library preparation, but then you could use a flongel, and I would say I think you could get a result within the same day if you would do it. But then also it kind of is the assumption that you have someone that's good with molecular biology, and that you would use a standard pipeline like something I mentioned, and you might not really care about an 8-hour workday, because and I also like you might, and that's assuming sequencing, but you know, if you have this is the kind of stuff that I know some people looked at illegal trading of eels. They use the qPCR. So if you just know what you're looking for, if you have an assumption which species it is, you have a reference, you can compare that. You know, if you have the right primers for a specific species, you could do a qPCR, and then that would be hours. So yeah, we can also talk about this in more detail another time. I'm happy to do that in more detail, because yeah, there are different ways to go. But if you want to sequence, I think you would say maybe a day, and you could do qPCR, but it requires you to actually prepare a species-specific marker, so to say.
That's great. Thanks a lot. I'd love to follow that up.
Cool. Let's do that. Nice.
Right. I think that's at the end of all the questions, and I think we've taken up enough of your time, Ineke. So thank you so much for that fascinating talk. I mean, based on the feedback just in the chat, it resonated and was super helpful for everyone. So much appreciated. And we'll share the link to your website and the call for contributors as well after the tutorial wraps. For everyone else, just a reminder that next week is our last Tech Tutors. Oh my goodness, I can't believe we've made it here. However, it is the start of a mini series, so it will go on for another 5 weeks, which I don't know how happened, but Akiba, of course, got excited. So Akiba and Jacinta are doing a big old deep dive into Arduino and building, guiding you through how to build open source data loggers for your project. So check that out. And Ellie's just dropped in the link to register. Otherwise, everyone else, we'll see you next week, hopefully. Ineke, thank you so much. And thanks everyone for a wonderful chat. And you know,
I just wanted to also thank everyone for showing up and for all the questions. It's really nice to hear that it resonated with people, and I'm just really happy that it's useful for people. So yeah, don't hesitate to contact me if you want more personal advice or you have more questions.
Oh my goodness. Well, that's an incredibly generous offer. Thank you. Everyone, if you want to contact Ineke, her contact details are in the data wrapper notes, so just check that out. Okay, that's a wrap, everyone. If you want to hang around, we normally have we stop recording and just have a little chat about tech and, you know, just I don't know, chat with each other, because we're all in tech together. So if you want to hang around, you're most welcome. Everyone is welcome, because we're a community. Otherwise, we'll see you next week. Thanks, everyone.